I am Priyanka Arora. I am from the Department of Biotechnology, Rama University. I am here to give the lecture on pyrosequencing. Pyrosequencing is a type of DNA sequencing. It is one of the methods of DNA sequencing. There are several other methods of DNA sequencing such as chemical degradation method, which is given by Maxim and Gilbert, dideoxy chain termination method, which is given by Sangers, the pyrosequencing method, and of course next generation sequencing method. Today I am going to cover pyrosequencing method. So what is DNA sequencing? DNA sequencing is finding the sequence at which the nucleotide is present, that is the order in which the nucleotide is present in a given stretch of DNA. If we look at the procedure, how the DNA sequencing is carried out using the pyrosequencing is, first we take a template DNA. The template DNA is a DNA strand which is to be sequenced. Okay. This template DNA is first denatured and we get a single stranded of the DNA sequence, the DNA stretch. Okay. This DNA is required to be PCR'd so that we will get the amplification of the DNA. If the DNA content is very less, we will increase the number of copies of the DNA by doing the PCR. To do the PCR, we know we require a primer, that is we require a free 3' OH. For the primer, and since we do not know the sequence of the DNA, for the primer we can clone this DNA in the vector. If we clone the DNA in the vector, the sequence of the DNA, the sequence of the vector can be used as a primer sequence. Okay. So, first this DNA is denatured, the single stranded DNA is taken. This single stranded DNA, it is coded in the magnetic beads. In the magnetic beads, this DNA is coded. Okay. This DNA is coded in the magnetic bead using the biotin label, and after coding it in the using this, we will keep this in the Eppendorf tube. In the Eppendorf tube, we will put the reaction mixture which is required for the PCR. Okay. The type of PCR which we do using for the pyrosequencing is the emulsion PCR. For the PCR, we will make the reaction mixtures. For that, we will add template DNA, we will add primer, we require DNA polymerase enzyme, we require buffer, DNTPs, apyrase, APS, sulfurylase, luciferin, luciferase. Okay. This is the reaction mixture we have to make before the start of the reaction of the pyrosequencing. Okay. We know what is the work of all these enzymes till DNTPs in the PCR, but these we will see one by one what is the function of all these remaining substrates and the enzymes. Okay. So, first when we have coated the DNA in the beads and we have taken all the reaction mixture in the DNA, we will see this is a single strand of the DNA, this is a template. If we will see what is the sequence of the DNA, suppose this is G, C, A, A. Okay. So, we will add DNTPs in the reaction one by one, knowing which DNTP which we are adding. Okay. If the DNTP gets bind to the primer, it means the complementary of the sequence is present in the DNA sequence. Okay. So, if we are adding in the reaction mixture DCTP, the DCTP will bind because it is complementary to the G. The binding of the DCTP here is by a phosphodiester bond. We are adding DCTP in a form of triphosphate, but the form in which it is incorporated is monophosphate. The alpha phosphate is incorporated in the C, rest the beta and the gamma phosphate is removed as a PPI, which is known as pyrophosphate. This pyrophosphate is measured. If there is a binding of the DNTP which we have added, then the pyrophosphate will be released. If this pyrophosphate is released, it should give a reaction such that a light is produced which is detected by the detector. Okay. So, how this reaction is carried out, we will see that. Okay. When a PPI is released in the presence of APS and enzyme sulfurylase, it will produce ATP. This ATP will further react with luciferin, the substrate which was present in the master mix which we have created, and in the presence of luciferase enzyme, it will produce oxyluciferin and also emit light. This light which is emitted is detected by the detector. Whenever this reaction has taken place, the light detected by the detector will be produced in a form of a pyrogram. Okay, so it has added C, it will make a note in the x-axis that the C nucleotide is added. Okay, if we have added 1 mole of DCTP, it will, and the reaction is given, it will make the amount of intensity of the light which is produced. Okay. So, in the next step, if we add another set of nucleotides, suppose if we have added T, it will make the account of the nucleotide which is added such that it has added T. But since the T will not incorporate here, it is not complementary to C, it will not produce PPI. The PPI not produced will not produce the light, therefore, there will be no signal in the graph. Similarly, in the next step, if we add DGTP, instead of if we add DGTP, it will incorporate here G and it will produce PPI and it will give the light signal and produce a signal. But in this reaction, the amount of DCTP which we are adding or the DGTP which we are adding, every amount of this nucleotide will not be used. To remove the excess of DNTPs which we are adding, in each step we use the enzyme apyrase. This enzyme removes the unused DNTP which is present in the reaction every time the reaction is completed. Okay. So, here the G is incorporated. Next, if we will add DTTP, then it will add to it. Okay. It will incorporate, it will produce the pyrophosphate, but the DT will be added twice because the next nucleotide required is also T. So, the intensity of the light produced will be twice as compared to the intensity of the light which was produced earlier. Okay. So, in this way we can find the sequence of the DNA which we are, the DNTPs which are getting incorporated in the DNA. So, there is one catch in this method is that whenever we are adding DATP as a amount, as a reaction mix for the reaction mixture, if we are adding DATP, this DATP can get confused with this ATP, and this DATP which we are added can react with luciferin and produce the light. Okay. So, this can give a false or early reaction and can give a false reaction that A is present. But so, to remove this false reaction, we will add ATP in a form of modified nucleotide such that we use deoxyadenosine alpha thiopyrophosphate, tripyrophosphate. This is a modified form of DATP which we are using in the reaction so that it does not get confused and give the early reaction with the luciferin and produce every light every time the DATP is added. Okay. So, this sequence, when we will see the sequence, it will be the amount of the nucleotide sequence which is incorporated in the 8th step. The original sequence will be the complementary of the sequence which is produced by the pyrogram. In this way, we can find the sequence of the unknown DNA using pyrosequencing. Thank you.